Hello everyone, in this video we will talk about structure of compound microscope. Look at this picture. Here we can see this is the structure of compound microscope, which is first invented by Z. Janssen or Zacharias Janssen. Now first let us talk about the different parts present in this compound microscope. The first, let us talk about this base. Weight of the microscope is held by the base. Okay. The next, the important part, this is called arm. Arm is used to hold the microscope. Just let me note down. Here base is the important part, and the next one is arm. With the help of this arm we can hold the microscope. And another important part of the microscope, see, this is body tube. In this body tube, a draw tube. See, this is the draw tube. This is body tube, and this one is draw tube. And another important point of this compound microscope is lenses. Here in this compound microscope, two type of lenses are present, and these two type of lenses are, one is present at top of this draw tube. So this one, this is eyepiece lens, which is also called as ocular lens. Eyepiece lens, also known as ocular lens, through which we'll see the object which is placed on the stage with the where we already prepared a temporary slide. And another type of lens is present at lower part of this body tube. In the lower part or posterior part of this body tube, we can see that there is a nose piece, this round one. This is nose piece, and to this nose piece, three objective lenses are present. So in compound microscope we can see the two type of lenses. One type of lenses or eyepiece lenses are also known as ocular lenses, and another one is objective lenses. Here more than two objective lenses are present with different magnification powers. Now let us see here the magnification power of eyepiece lenses ranges from 5x to 10x and 45x, whereas the magnification power of objective lenses would be also 5x. Its range is from 5x to 10x. It will also be 45x and 100x. Not all the objective lenses with the same magnification power, they are with the different magnification powers, right? And another important part of this compound microscope is the stage. Just below these objective lenses you can see that there is a square shaped structure, stage. Here on the stage clips are present. So we can call them as stage clips. Stage clips. These two stage clips are present. See, this is one stage clip, and the another side there is one more is present. These two stage clips are used to hold the glass slide. Hold the stage clips can hold the glass slide. Next, just below the stage, there is another important point. So we call it as condenser. In this condenser, we can see there is a presence of diaphragm lever, and there is a slit present which is passes through this stage. Below this condenser, a mirror is present. So this mirror acts as a reflector. It acts as the reflector. So whatever the light fall on this mirror, that will be reflected and that passes through the slit present in the stage. And after preparation of the temporary slide or a permanent slide, if we wanted to observe any plant cell or animal cell or any other objectives that we need to prepare a slide, or permanent slides are also available. So when you place it on the stage, see, with the light which is reflected by the mirror will passes through that material which is placed on this glass slide. So that image can be seen with the help of these objective lenses and eyepiece lenses. That image will be formed in this body tube. Now let us talk about the important parts of this compound microscope. The coarse adjustment. This is rotated in both the directions. So when we are rotating it, the entire body tube along with the draw tube and objective lenses can move up and down. So here this can be moved to get the desired focus. To get the desirable focus, we will rotate this. Whereas the fine adjustment can also be rotated in both the directions. This can be rotated in both the directions to get the sharp image of the material whatever we have taken on the glass slide. Just let me note it down. The coarse adjustment is used to get the desired focus, whereas fine adjustment is used to get the sharp image. Now let us talk about working mechanism of compound microscope. The working mechanism. Whenever we wanted to observe any plant or animal cells, so we have to prepare a temporary slide, and when you place it under compound microscope, we have to fix this on the stage like this with the help of stage clips, so that the glass slide cannot move here and there. So here when the light is fall on this mirror, the light will be reflected and it passes through the material which is kept on the stage. And here with the help of objective lenses and ocular lenses, we can see the image. So here actually the real and inverted image of this material which we have taken on the glass slide is formed by the objective lenses. So here objective lenses forms the real and inverted image, and it is further converted into virtual image by the help of these eyepiece lenses. So this is the working mechanism of the compound microscope. Now just let me note it down. The real and inverted, the real and inverted image, the real and inverted image of the object formed by the objective lens is changed into an erect and virtual image, an erect and virtual image by the eyepiece lens. So this is about structure of compound microscope and its working mechanism.